Okay, Marcella, I'm going to do my best. You've given me a lot of questions here. So muscle contraction when ATP binds to myosin heads. So basically, muscle contraction is based on something happening in sarcomeres, which are these units, which are not cells, because they don't have a nucleus. Okay, and you have these elements that interact. Those elements being myosin, which is a thicker protein, and actin. And what's happening is these elements are sliding past each other. And that sliding past each other causes each one of these very small units called sarcomeres, this is a sarcomere, to contract. Okay? If a sarcomere contracts, if all the sarcomeres contract together, I mean the whole muscle contracts. That's how it works. So the question that you're asking is related to how they slide past each other. How does actin and myosin slide past each other? And they slide past each other, we draw them kind of large. Because myosin has these heads that attach to the actin. And the heads move. They cause the actin to slide. Okay? So specifically what does ATP do is it kind of moves the head to a position where it is ready to move. So these myosin heads are actually moved back. They're kind of what we call cocked, which is a word that comes from the world of guns. Okay? Because guns have a revolver type gun has a little thing called a hammer. And that hammer has to be cocked in order for the pistol to fire. So you pull this thing back, it's cocked, and then when it fires, the gun shoots. And that is a little bit like the energy is being stored. And what ATP does, of course it becomes ADP because it expends some energy, is it moves that myosin head into a ready position. So it's going to bind to the actin, and then that energy is released and it causes the actin to move. Bend it back, that's ATP, attach it to the actin and then release, and it pushes the actin along. And this happens many, many times. Okay? Good. Let's look at the next question. So osmoregulation. Osmoregulation is the control of salts and water in the blood, because your body needs to keep a bunch of things the same in order to be stable. And salts and water, okay, together, if I'm controlling these two things together, what I'm controlling is called osmoregulation. Okay, why? Because osmo comes from osmosis. Okay, and osmosis is the movement of water in and out of cells according to the salt concentration. So your body trying to control these two things together, that's osmoregulation. Right, next question, the function of the loop of Henle. So let's go to the loop of Henle. Loop of Henle is that big long loop in a nephron, which is that tube which occurs inside kidneys. There we go. And if I show it untwisted, which is the most simple way, because the reality, the loop of Henle is twisted. Right. The loop of Henle is this bit here. And what is its job? Its job is osmoregulation. So well done. Okay? Osmoregulation is exactly what the loop of Henle is for. It's for absorbing water and salts. Okay? Especially water. How does it do this? Well, this part here absorbs salts. Now the salts gather in this space. We call this space the interstitial space, the space in between. And that increases the salt concentration, which attracts water. So the water comes out of this side because there's more salts, and water is always attracted to salts. So as the water goes down, it starts being attracted out of the nephron into the interstitial space because there's salts accumulating in the interstitial space, which is coming from this side of the nephron. So the longer this nephron, the more effectively you can absorb water. And you'll see desert animals have a really long loop of Henle. This, by the way, because it goes this way along the nephron, this is going down, this is going up. So this is called the descending. Okay, arm. This is called the ascending arm of the loop of Henle. Great. Okay, what's your next question? ADH. So ADH is a chemical made in the brain, antidiuretic hormone, which tells you if you're becoming too dehydrated. So let's imagine I haven't drunk water all day. I've been drinking caffeine, which stimulates urination. It's one of those chemicals which is associated with making the kidney excrete more water than is necessary. I'm going to get dehydrated. So if I've been drinking a diuretic like caffeine, then I'll be going to the bathroom a lot and I'll need more water. So if I need more water, how can the brain do something about this? So what the brain does is a part of the brain called the hypothalamus. Okay, so you've got your brain, right? Lovely organ, there it is. Somewhere here, there's the hypothalamus, which is detecting and monitoring everything in your body, the whole situation, kind of what changes. pH, water content, salt content. And it says, you know what, we're getting kind of dried up. What are we going to do? Well, it makes this hormone, ADH. Well, specifically what it does is it tells the pituitary gland to make ADH. So the pituitary gland is told by the hypothalamus, make ADH, we're getting dehydrated, so it makes ADH. What does ADH do? It travels in the blood and it goes to the kidney. And when it gets to the kidney, it causes the kidney to reabsorb more water. So more water, it makes this part two places. Here, and I'm going to put a plus sign because that's better than that. Plus H2O. You can absorb more water, and also here, they're collecting that. More water. So you're reabsorbing more water, which means you're saving water, which you would have lost otherwise. This has the effect of making your urine more concentrated. You can notice that if your urine is looking darker in color, more orange, that means you're probably dehydrated. But what's happening is your brain is making your urine more concentrated by telling your kidney in this part and this part to absorb more water. What are these parts? This is the collecting duct. And this is the distal convoluted tubule. Okay, I've still got a gun on the board. That's a bit strange. That's that question. Okay. Whoops. Sarcolemma. So sarcolemma, if you look at muscle structure, imagine I'm flexing a muscle right now. You've got a muscle, let's say it's a bicep, okay? But the tendons, it's attaching to a bone somewhere. Okay, we've got this bicep here. I haven't drawn the attachment accurately, but it doesn't matter. Okay, if you look at this, then you'll find it's made up of muscle fibers. Which are these cords which we tear when we play sport. Okay, these long rope-like things, muscle. Right, if you take one of these muscle fibers and have a look at it, you'll see that the muscle fiber is actually covered in a membrane. And the membrane it's covered in is called the sarcolemma. We'll notice that it has nuclei dotted in it. So there's a big nucleus every now and then, because it's not divided into cells. Okay, it just have nuclei, and it's also made up of smaller fibers.
So the fibers are made of fibers, kind of like when you look at a rope. And the fibers are called myofibrils. Now the thing which is important about the sarcolemma is it delivers the calcium. Because what makes muscles contract is calcium. Every time you do anything, move my arm, and moving my arms, calcium is being released to my muscles to cause those muscles to contract. How does the calcium get there? It gets there because the sarcolemma delivers it. Okay, that's that. Right, functions of the lining of the small intestine. So you're looking at the small intestine. You know that it starts after the stomach. You've got a little loop here. First 10 centimeters, of course, is called the duodenum. And then the rest of it, which goes for 9 feet, is the ileum. And the ileum is wonderfully coiled up in our body, so we don't realize how long it actually is. So this first bit, the duodenum, is more for digestion. And this bit here is more for absorption. And the reason this is because the duodenum receives secretions from the pancreas and the liver. But all of it has a lining which is designed for absorption. So the lining is designed for absorption. And it's designed for absorption because these wonderful villi, okay, with all their associated features that they have. For example, they have a single cell. They've got lacteals in the middle. They've got blood vessels. All to make absorption effective. Now, what's the difference between the duodenum lining and the lining of this part? Well, you've got some glands in between here. And the glands can secrete some enzymes. Because the lining of the small intestine also secretes enzymes as well. Okay, and you get more of the glands in this part because that's more where digestion occurs. You do get some glands in the ileum. Broadly speaking, the lining of the intestine is the same throughout its length. There's just a few more glands in the duodenum because that's where you expect to have more digestion. Okay. Right, 2 kinds of white blood cell, phagocytes and lymphocytes. Basically, right, you've got big old phagocytes, which are cells which eat bacteria. So they'll go around the body and they'll find a bacteria and they'll engulf it, which means that they kind of eat it using their cell membrane. And it ends up inside. Once it's inside, it gets destroyed by a special, basically, destroy anything, powerful enzyme bag called a lysosome. Anything which is bad in a cell can be sent to the lysosome and it's like death, it's destroyed because it's a really powerful enzyme. And that's what happens to bacteria. These are phagocytes and they're what we call parts of the non-specific immune system. But basically they just eat anything which is not part of your body. Anything that doesn't belong there. It could be a splinter in your finger, it could be a little piece of sand. They eat anything. And that's one of the main 2 kinds of white blood cell. And then you've got lymphocytes. Okay? Lymphocytes are smaller. And by the way, I've drawn a specific kind of phagocyte here. This is what we call a macrophage. Because there's actually more than one kind of phagocyte. That's a very common one. Okay? If you look at a lymphocyte is very different. What it does is it makes these Y-shaped specialized proteins called antibodies. And those are chemical weapons that can affect bacteria. They can trap them, or they can cause the bacteria to stick together in clumps. They can kind of round them up. They can get in between them and cells. They can stop them from entering cells because a lot of viruses particularly get their way by entering cells. All those things that they do. They can stick to bacteria and make the bacteria more noticeable. They do a whole bunch of things which which could be, so they could be there blocking the bacteria from getting into cells. They do all kinds of things to protect us. And those are the two main kinds of white blood cells. Of course, I mean, there are other kinds of white blood cells. For example, T helper cells are a kind of lymphocyte, and they just help. They don't actually make antibodies. And then you have the B plasma cells. And those can be activated to become B cells by a specific antigen, and they make a specific antibody. And it's specific, right? Because what we're talking about here is a specific response. I have this COVID-19 or whatever. Let's not talk about COVID-19. I have this hepatitis virus. I need the hepatitis antibody. So you get the hepatitis vaccination. That's one way of getting the antibodies. Or you could get sick with it and you let your immune system deal with it and respond. And that's what it does. So, let's just move the board. Make sure I've captured all of that in the frame. Okay, so T helper cells help. What do? They alert. They send messages. They communicate with the B plasma cells. And the thing about T helper cells is HIV destroy them. So HIV take out the T helper cells, which means they're no longer communicating with the B plasma cells. If they're not communicating with the B plasma cells have less help. They're less likely to make antibodies in time, which is why HIV is such a serious illness. It makes you vulnerable to everything else. Your immune system is not communicating with itself as effectively. Right. I'm getting there. How to interpret a spirogram. Basically, it looks like this. Breathing goes here. When you breathe in, it goes up. This is volume. This is time. I breathe in, I breathe out. I breathe in, I breathe out. Okay, if I breathe fast, rapidly, they get close together. If I take a big breath, it gets bigger that way. Okay, that's how you interpret a spirogram. Now, I can get some measurements from it. If I take the difference between any of these peaks from there to there, that difference, okay, which is delta V or delta volume, then I can tell the volume of this breath. For example, if this value is 700 milliliters, because it has to have a unit, and this value here is at 200, then delta V is 500. Which is 700 minus 200. Another thing I can do with a spirogram is I can calculate breathing rate. Let's say this is 30 seconds. I can say there's 1, 2, 3 breaths in 30 seconds. So 3 breaths, 30 seconds. I can say, well, there must be 6 breaths per minute, which is twice 30 seconds. Okay, so you can do that with a spirogram. You can also guess what it would look like if people take certain breathing patterns. Emphysema. So we're still talking about breathing. This is kind of related, but some people they have to breathe a lot. Okay, it's like they're breathing a lot to do something simple. They're climbing the stairs and they have to breathe huge breaths. They're like this breath there, big, close together breaths. So other people don't need to. And that's partly to do with personal fitness, but it could be to do with something called emphysema. Emphysema means that people need to breathe a lot. They pant, and it's because that their lungs aren't efficient. So why their lungs aren't efficient is because if you take the lung structure is basically made out of these alveoli, which are little air-like sacs. Okay? So if you looked at a lung under a microscope, if you imagine a human lung, imagine the human lung, a trachea, and you took a section of it, it would be made up of these little air sacs called alveoli. Now, people who have emphysema, the alveoli have broken. So there's actually, instead of having lots of small alveoli, they've got a couple of big spaces. And what this means is there's less surface area, because all of these lines represent a boundary between air and blood. okay, because there's blood in the lining and it can be absorbed. So there's less surface area. If there's less surface area, that means they get less oxygen. You get less oxygen, basically they have less ATP, and ATP is the bottom line, it's energy. They don't have enough energy, so they have to really work hard and breathe much harder than us to get that energy. How can you get emphysema? One of the classic cases is smoking, okay? Particularly direct smoking, smoking yourself. Smokers very often suffer from emphysema because the compounds in smoking destroy the lining of alveoli. Okay, great. We're almost there. Neonicotinoid pesticides. So what neonicotinoid pesticides do right, they work here. You've got a synapse. Synapse is a place where you pass an impulse from one synapse to another. So you've got the pre-synaptic cell here, or the membrane, which is the end. You've got the post-synaptic membrane. And the impulse has to somehow jump this gap, okay? This gap which is called the synaptic cleft. Okay, so what often happens when you do something, okay, like I catch this pen, is I have to jump between various synapses to send that signal. I have to jump through these spaces. And of course, we know that what happens is there's neurotransmitters, and those neurotransmitters are released, and they carry the signal across the synaptic cleft. But what neonicotinoid pesticides do is very interesting, because once there's a neurotransmitter called acetylcholine, and it's used a lot in movement, okay? Movement in nuts, especially movement in insects, okay? And it carries a signal across circuits involved with movements of the body. Now, after acetylcholine has been used, it needs to break down, because you can't leave it there. It's broken down, break down, and it becomes two chemicals, acetylcholine. Right? How do you break down a neurotransmitter? You actually use an enzyme. Enzyme. To do anything in the body requires an enzyme, chemically speaking. This one has a name which makes sense. It's called acetylcholine esterase. So basically, it breaks down acetylcholine. Now, what neonicotinoid pesticide does is it takes out this enzyme. Neonicotinoid, okay, block the enzyme. Okay, that's the enzyme acetylcholine esterase. If you block this enzyme, what happens? This stuff doesn't break down, it stays there. It stays in the synaptic cleft, it continues to stimulate the signal. And you keep getting the signal even though that you don't. And what makes you move is the difference in signal. It's going from not having a signal to having a signal. And so if you keep having a signal, it's the same as not having a signal. There's no difference, which means basically paralysis. Paralysis and death. For humans, less so, but for an insect, definitely, because insects are really small, much more vulnerable, and they have much more acetylcholine. Okay? And that is the answer to all your questions. I need a coffee now.